I'd like to first introduce our first panelist, David Chen from Biochemistry. David's presentation is on enhancing qPCR detection of Candidatus Liberibacter asiaticus. I may have mispronounced all of that, but my apologies. And David, feel free to correct me when you do your presentation. David, just for a bit of a bio here, is from Biochemistry, works with Dr. Gregor Blaha in Biochemistry, working on the same project. David is also a senior member of honors and conducted research under Dr. Blaha's lab since winter of junior year, and is now planning on taking the MCAT in June and pursuing medical school down the road. So please give a warm welcome and applause for David Chen. David, feel free to take it away.
All right, thank you. I would just like to introduce myself again. My name is David. I'm a biochemistry major, and I'll be presenting my project on enhancing the qPCR detection of Candidatus Liberibacter asiaticus. So here is an outline of what I will be presenting today. So I'll be starting off with what exactly is Candidatus Liberibacter asiaticus, and then what disease it causes. I'll be presenting my goal for this project, and then I'll be presenting my methods and my results, conclusions for my method 1, and conclusion for my method 2. So let's get started. Candidatus Liberibacter asiaticus, or CLAS for short. What exactly is it? So CLAS is a gram-negative, phylum-limited alpha proteobacterium that falls under the genus class of Candidatus Liberibacter, and other species of Candidatus Liberibacter are Africanus and Americanus, but for this presentation, I'll be focusing only on the asiaticus form. And what Candidatus Liberibacter asiaticus causes is a citrus-associated disease, also known as Huanglongbing, HLB for short, or it can also be called citrus greening. And the main way this disease is transmitted is by this Asian citrus psyllid that you can see in the image in the bottom right. So what exactly is HLB, or citrus greening disease, and why is it important? Well, HLB was initially discovered in South China in 1943, and it develops symptoms. It has several stages of symptoms, resulting in stage 1, where the leaves of the host plant become yellow and blotchy. Stage 2 actually leads to the reduction in fruit production and quality that results in bitter tasting fruit. As you can see here in the image, the fruit doesn't taste that well, look that well. And stage 3 actually eventually leads to the death of the host plant. And why is it important? Because it's currently the biggest threat to commercial cultivators of citrus to this date. And being in Riverside, we are known to have a large citrus grove. So this disease has a major impact on it. So my goal for this project. So my goal is to try and improve the detection signal of the pathogen during quantitative PCR. So what exactly is qPCR? qPCR is a molecular biology technique that allows quantification of specific nucleic acid sequences during replication in a sample. So some of the components in this reaction involve the ITAC universal probe supermix, the primers, forward and reverse, the template, and the hydrolysis probe. So the hydrolysis probe is an oligonucleotide sequence that contains a fluorescent reporter dye, also known as a fluorophore, and a quencher dye. So why are we using qPCR? Because previous research has shown that culturing of the bacteria has been proven difficult because of specific reasons. Those reasons being that the bacteria is actually obligate in nature, meaning that it cannot survive outside of a host. and it requires the nutrients that the host plant has in order to survive. It also reproduces very slowly, so it doesn't generate as many copies of itself as itself. And it also lacks a cell wall. So because of its cell wall, it cannot survive the harsh conditions of the outside world. So how am I going to accomplish this goal? So my methods are trying to use RNA instead of DNA to try and improve the signal detection during replication, and also using the addition of proteins to help enhance the signal detection as well. So let's start off with method 1. So using RNA instead of DNA. So by using reverse transcriptase, RNA can be converted into cDNA or complementary DNA and used for qPCR. And this allows the sample to contain more nucleic acids instead of just DNA itself. So having the total nucleic acid of RNA and DNA combined is more than just DNA itself. And we found that the concentration of your nucleic acid sample and the cycle threshold value, or CT value for short, are inversely proportional, meaning that the more concentrated or the more nucleic acids you have in a sample actually leads to lower CT values, which is what we want in this project. So the procedure, how I'm going to do, or how I'm going to accomplish for method 1 is I would extract both RNA and DNA from the same infected tissue sample using a Dianese plant mini kit and its protocol on extraction. So I would have one sample that has RNase present, while the other sample has the absence of RNA. The sample with RNase present will only have DNA remaining, meaning that the RNase is a protein that degrades RNA, of course. And the sample without RNase will have our total nucleic acid, which includes both RNA and DNA. And I'll showcase my results for the first method. So using the observations at the extracted sample at high concentrations of nucleic acid, we can see that the difference between RNA and DNA was roughly a 1.5 cycle threshold difference between the two. And when we took a t-test to see if this result was significant enough that the two samples are not similar, we found that the probability of our t statistics was 0.003, and which is lower than our alpha that we set at 0.05. And because of this, we can say that it is statistically significant that these two samples are not similar. And here is just a table of the calculated results of the t-test. We can see that our t-stat is -57 and our t-critical value is 4.30. And with that, we can, with the probability that our t-stat is greater than or equal to that than our t-critical is 0.003. So I decided to expand on that experiment and test whether it had an effect on low concentrations of nucleic acid. So I took that initial sample that I did with the previous experiment, and I did a 1,000-fold dilution of that sample and ran the qPCR reaction with it. What we notice is that with RNA, we actually do produce all three signals that we initially started with in each replicate. And with DNA, we're actually missing some of the signals, which means that RNA is actually producing or performing better than DNA in signal production. So my conclusions for method 1, the effects of RNA demonstrated that it performed better than DNA, and improving detection of signal with, it also improved the detection of signal with low concentrations of nucleic acid, as you saw in the previous graph. And this actually opens up a new path to experiment with the limit of detection capable by the PCR system. And some of the real world applications that we can use this experiment on would be to allow us to detect suspected infected hosts before the infection spreads to other healthy other plants. And this suggests that it's possible to detect less than one molecule of DNA in a single cell when we're using RNA instead of DNA. So I'll move on to my method 2, which is using protein additives and to try and enhance the detection of our signal. So proteins are known to aid in replication of DNA by either increasing processivity, stabilizing, synthesizing, proofreading, repair, regulating, and terminating. So the 3 proteins that we tested in this experiment are T4 gene 32 protein, RecA, and extreme thermostable single-stranded binding protein. And for T4 and extreme thermal ET-SSB, it is known to increase possessivity of the DNA polymerase in a qPCR reaction, while RecA enhances the primer template binding. So results of using the stock solution of our protein that we obtained, we noticed that there was no signal being produced by either T4 or ET-SSB. And the only signal that was produced was from RecA, but as you can see, the CT value of RecA is actually much lower or much higher than that of our control of water and buffer, which is not what we want to see. So I decided to do a titration series of the original stock solution, thinking maybe it was too concentrated and it's actually inhibiting the qPCR reaction. So after I did the titration series, I still saw no improvement with the protein additives, but I did notice that as the concentration of protein decreases, the CT value actually becomes closer to that of water to that of our control of water. So as we can see here, as the concentration decreases, we can see that the CT value becomes closer and closer to water. So we did a little revision to our method 2. So we actually found that there was 50% glycerol in the storage buffer of our protein. And this is to avoid freezing when stored in the -20. So we also know that glycerol does inhibit qPCR reaction. In a later slide, I'll show you the results of different glycerol concentrations affecting qPCR, the reaction. And we decided to try and remove this glycerol in the storage buffer by doing a dialysis using a 225 micromilli filter paper. And we tried to exchange the buffer with a 2/3 ratio of tris saline buffer and water. And the reaction mixture is similar to that of with a regular qPCR, but the water component of the reaction mixture is actually replaced with the dialyzed protein instead of water. And for the water control, it would still be used as water. So here's the glycerol test for our, here's the glycerol test that I tried with different concentrations of glycerol and its effect on the reaction. We can see that with higher concentrations of glycerol, it actually inhibits the qPCR reaction because as you can see here, with 30% glycerol in the reaction mixture, there was no signal being produced. And depending on the glycerol concentration, you can see that sometimes it's inconsistent with signal production and the standard deviation of the signals are very wide. So we know that glycerol inhibits the reaction. So after doing the dialysis, I ran the tests again with the same concentrations that I used with the undialyzed and tested if there is any improvement in CT value. From the results that I obtained, again, I saw no improvement of CT value compared with the control of water and buffer. And we do see that as the concentration decreases, it does mimic more similar to water due to the fact that because it's so diluted that the protein is non-existent at the point. So it's pretty much water at that point. So I extended this experiment against like the RNA where I tested low concentrations, and I did see that with the low concentrations of DNA, water and buffer did have trouble producing a signal, whereas some of the other proteins such as RecA and ET-SSB also have trouble producing signal. But there was one signal that appeared, which was at 0.05 micrograms per microliter at T4 of the T4 protein, and both replicates produced a signal. So to test if this was the case, I decided to do more replicates of that concentration, and it turns out that, again, control of water and buffer still have trouble producing the signals at such low concentrations of DNA. And we also see that with the T4 protein, it produced signal, but it also was having trouble producing all of the signals for all 5 replicates. So it wasn't really sure that if this concentration of T4 was actually enhancing the detection at such low concentrations of DNA. So with my conclusion, I found that the effects of protein with the undialyzed showed inhibition to the qPCR reaction due to the presence of glycerol. I also found that as the protein gets more diluted, it becomes more like water, so it mimics the CT values of water, but doesn't really improve the reaction. There was no improvement after dialysis. All the CT values of the protein were higher than that of the water and buffer control, and improvement of the detection at low concentrations of DNA also revealed inconclusive results. And it's highly likely that it's up to random chance that the signal actually gets produced. So my reasonings for why the DNA sample could still be inhibited prior to dialysis and after dialysis would be because of the presence of glycerol still in the storage buffer even after dialysis. So hints of glycerol is actually still inhibiting the reaction. And this glycerol actually inhibits the primer, probe, and DNA polymerase from functioning efficiency due to the increase in viscosity of the solution. And the presence of high salt, because we use a tris saline buffer, can also inhibit the DNA polymerase or interfere with the fluorescent detection. And it can also precipitate out the primers and probe, resulting in failure of the qPCR. And with that, that concludes my presentation. Thank you for listening, and I'll open up the floor for questions.
All right. Thank you, David, for that wonderful presentation on your research. Yes, let's give a hand or an emoji hand, clapping hand for David. And yes, let's open it up for a brief Q&A, and we will have a last Q&A after all the presenters go as well, should any questions arise, right? So, and